In this video, we will discuss about nitrogen fixation in the atmosphere and also within the plants, and then understand one of the application of this nitrogen fixation related to dieting. So let us start with the nitrogen cycle in the atmosphere. So what happens is let us start with the nitrogen that is available in the atmosphere. So this nitrogen fixation occurs. It means that this nitrogen that cannot be utilized in its native form is converted into, or is been fixed by the bacteria, or it's converted into some compound such as this ammonia, and then it can then be used as a nitrifying bacteria that can convert this ammonia into the uh, nitrogen uh, nitrogen trioxide. And same thing, this ammonia can also be come from the other sources such as from the uh, from the ground level, because of nitrogen fixation at the ground levels, and this will also finally lead to form a nitrifying bacteria by some process for that will convert this NH4 into the NO2, and then nitrification will lead to this nitrifying bacteria. And after that, this denitrification of the bacteria occurs, and during this denitrification the nitrogen will again release to the atmosphere, and this nitrogen will then again go follow the same process. This is called the nitrogen cycle process. So how nitrogen fixations is related to or happen in the human beings is that this nitrogen will come into will go, or we can say will come into the body of the animals or bacteria through diets, and these diets should be in the form of protein, amino acids, and this will finally leads to formation of proteins within the body, and this occurs one of the important compound that leads to conversion of this proteins, amino acids into the human proteins is the glutamate. The presence of glutamate is usually found in uh, many mammalian cells, many mammalian organisms such as bacteria, human beings, and this glutamate will convert this nitrogen nitrogen containing compounds into the final human proteins that can be utilized for the normal functioning of the body. And glutamate is uh, this glutamate does not uh, do its work independently. However, it requires the help of glutamine and alpha glutarate, as you can see in the figures, that if glutamate is converted into the alpha glutarate, then this alpha keto acid will help in the conversion of this glutamate formed product into the human proteins or the amino acid, alpha amino acids, with the help of aminotransferase. So that can finally leads to this formation of human proteins. These amino acid then can be utilized during translation process of mRNA into the structural protein. And so this is how nitrogen fixation occurs within the human being. Now we will understand one uh, interesting application, or interesting thing occurs during a specific form of dieting, that is fasting. So that things occurs is called glucose alanine cycle. So let us first understand what happens in the normal condition. This is the muscle cells, and this is the hepatocytes, or you can say a condition in the liver. So what happens in the normal case is that the glucose comes and glycolysis offers, that leads to the formation of pyruvate, and this pyruvate then pyruvate is the final product of when the glucose is final product of glycolysis of the glucose. So in the normal condition, what happens is that this glycolysis will utilize the nitrogen that is required for this process, and will discard the excess nitrogen again back to the uh, glutamate, as glutamate can act as both as a donor and acceptor of the nitrogen. So however, during the fasting condition, what happens is that this most of the we can say many proteins in the muscle cells, skeletal muscle cells, will degrade itself into the alanine, as it is shown here. This pyruvate that is usually that is in normal case being utilized for the functioning of this muscle cells. However, in case of fasting, the degradation of these proteins in the muscle cells will lead to the formation of degradation of the muscle protein occurs, and most of these muscle proteins degraded into the alanine, as it is shown here, with the help of this enzyme that is ALT, and the byproduct is also formed, that is lactate. However, our main focus is on the formation of alanine. So in case of fasting, the degradation of the muscle cells protein occurs that leads to the formation of alanine with the help of ALT, and this alanine will then move into the blood blood vesicles, where with the help of blood it will move into the liver, and in the liver what happens is that with the help of alanine, this with the help of this ALT, it will again be converted into the pyruvate, and this pyruvate will then be degraded. What happens in the in when aniline has been converted into the pyruvate, then this pyruvate can then be utilized. The energy, we can say, the energy, that is 6 ATP, with the help of pyruvate and 6 ATP, this gluconeogenesis process can occur, that again leads to the formation of glucose, and it will then again recycle back into the human body with the help of blood vesicles. So, so in the case of fasting, this pyruvate then first converted into the alanine and then moved into the liver, where liver helps converting into this alanine into the pyruvate, and this pyruvate again converted into the glucose. And what happens to the additional amino acid that comes along with the aniline? When the aniline converted into the pyruvate, the glutamate that has been left over from this the NH4 has been removed, and it will then, this rest of the part will then contribute to the urea cycle with the help of 4 ATP. So this is how the cycle occurs in the case of fasting. uh, in technical term it is known as glucose alanine cycle. So I hope you have understood uh, somewhat what is going on whenever a person does fasting. If you need to understand this thing in details, you can go through the references that has been provided in the description. That's all for this video. Thank you for your time.